Welcome, everyone, to Wild Farm Alliance's Whole Farm Sustainable Pest Management webinar series. Whole farm pest management is a way to fine-tune the many influences that affect the food we grow. It requires understanding ecological principles, paying closer attention to crop and soil health, more patience working with natural biocontrol systems, and better management of biorational and other pesticides. With this webinar series, we are lifting up the voices of researchers, ag extension professors, PCAs, scouts, farm managers, conservationists, and other agriculture experts, over 24 in all, to bring knowledge to you to make better production decisions. This will help as pest control products are being more regulated. The marketplace is demanding more sustainable, organic, and regenerative practices. And besides growing healthy food, you will help support pollinators, beneficial insects, birds, and soil microorganisms, and help to slow climate change. I'm Joanne Baumgartner with the Wild Farm Alliance. You can see our Farmland Wildways logo on the bottom right. With this, we envisioned hedgerows to the moon and back, which is about 500,000 miles of hedgerows, what the UK used to have before industrial agriculture. And what we'd like to see, lots more hedgerows, other habitat as well is beneficial and provides so many services to the farm. I'd like you to consider attending all 12 of the lessons in this course. We have 2 lessons per month through March 2025. And while you don't have to attend them all, we hope you do. Besides this topic, optimizing beneficial insects and their habitat, which is the lesson for today, we have 11 other lessons like habitat that offers a blanket of attack, managing pests with biopesticides. And all of these lessons are on Tuesday at 11 a.m., except for the 2 in November, the augmentative biocontrol one and the infield and edge habitat. Those are both on Wednesdays instead of Tuesdays. Other lessons are managing soil-borne pathogens without chemicals, beneficial birds and bats, plant health fosters pest suppression, soils beneficial entomopathogenic microorganisms, habitat for predators and parasitoids, whole farm scouting, and whole farm system thinking. As I mentioned, write questions in the chat and we'll make sure the speakers answer them after they give their talks. We'll also be running a couple of interactive polls midway through. And if you are wanting DPR credits, make sure and take those polls. They're required. And we'll have time to discuss the answers to those polls and more time at the end of the presentations for questions. At the end of the session, we'd love to get your feedback through a quick survey. If you want DPR and PCA credits, you'll need to give us your license numbers. And for DPR specifically, you'll need to take a quiz with 10 questions at the end and get 70% right. So in today's lesson, we have Dr. Nate Hahn, Assistant Professor of the Department of Entomology, the University of Kentucky, and Dr. Houston Wilson, Cooperative Extension Specialist at UC Riverside and Director of the UC Organic Agriculture Institute. Nate is going to start us off with his presentation.
Yeah, well, hi, everybody. I'm really glad to be here. I'm excited to talk to you about one of my favorite topics. And what we're going to do here, I think, is I'm going to have the floor for 15 minutes or so. And I'm going to give you some really kind of general 101 kind of information so that we all have like a broad, firm understanding together. And then when we pass things off to Houston, I think Houston's going to get into a little bit more specific stories about pests than I will. But hopefully I'll give you kind of a broad, basic framework to start from. Hopefully not too basic, but we'll see. So great. When we talk about beneficial insects, sometimes we're talking about pollinators, sometimes we are talking about natural pest suppression, and that's what I want to focus on today. So what is natural pest suppression? We better define it. It's when natural enemies suppress herbivores or pests and then release plants from herbivory and increase how much they can produce. So I think of that, since I'm an ecologist, I think of that as what we call a trophic cascade, where the third trophic level, the natural enemies, are eating herbivores, and those herbivores are eating the plants, which if you take the idea that the enemy of your enemy is your friend, then those natural enemies are friends to plants because they're suppressing the herbivores that are their enemies. Another way to think of it is that's autonomous pest prevention provided by nature. There was a study several years ago now where they did back of envelope calculations and tried to figure out how much money does natural pest suppression contribute to the US economy every year, and via avoiding crop loss, and found it's probably, I adjusted this for inflation, something like $7 billion a year. I'm sure that number is wrong. I would guess it's an underestimate, but it's a big, big number, right? So this is an important process we're talking about here. And if we want to understand what beneficial insects need, there is a clever acronym that can help us make a framework for that, and that is SNAP, which stands for shelter, nectar, alternative prey or hosts, and pollen. So we can go through those and look at this horribly anatomically incorrect beneficial insect that I found as a as a stock image. So let's go through the SNAP acronym. And the reason for that, why we need to think about that, is because a crop habitat on its own, especially like an annual crop habitat, but really any crop habitat, it's not all that favorable for natural enemies, right? Some are better than others, but relative to a wild ecosystem, it's not all that favorable. It's relatively homogeneous, right? Look at this picture of a cornfield. There's not a lot of heterogeneity, not a lot of variation there. It's pretty homogeneous. They're not that biodiverse, right? So within this field in this picture here, I can identify one species, which means most of the herbivores that are there are just things that would eat corn. It's a simple system, right? Not very biodiverse. And it's also frequently disturbed, right? So there's mechanical disturbance that goes into tillage and mechanical cultivation. There's chemical disturbance if there are like insecticide or fungicide or herbicide applications going on. So all these things make crop habitats not all that great for biodiversity on their own. So we need to think about what insects need and how to incorporate them either within fields or maybe next to them, right? So the first letter was S for shelter. So you can think of shelter as in different ways, really, but somewhere for beneficial insects to go when fields are fallow or when they're mostly bare ground. So I'm in Kentucky. I'm from Michigan originally, where there's a whole lot of corn. And if you watch a cornfield throughout a whole year, there's only corn on it for like 5 months. Then there's 7 months of, hopefully cover crop or something else, but often just bare soil. So they need somewhere to go when the field looks like that. They may also need microsites that are thermally better for overwintering. So they need a place maybe that's a little bit warmer, something like that. Maybe they overwinter in like thatch of perennial grass or behind tree bark, something like that. So those are ways to think of shelter. Another is that shelter is a place where there are residual source populations that are in refuge, so that when there's an event that happens in a crop field, like a tillage event or a pesticide usage or something that kills a lot of insects within the field, that there's a reservoir of insects that were sheltering in a different habitat that can then repopulate and fill those niches again. So those are kind of 3 different ways that I think about shelter for beneficial insects. The picture here I have was built as an example of a beetle bank, which is carabid beetles or ground beetles, which are voracious predators in some cases. They often like to have a perennial grass shelter area. That can happen at different timescales. It could be an overwintering area, or it could be where they go at night and then they venture out into the field to feed, or vice versa if they're nocturnal. Nectar is where beneficial insects are getting sugars, glucose, fructose, and sucrose. That's what they're using mostly for energy, as fuel to fly around and do the other things that they do. Nectar, of course, comes from flowers, but there are also plants, lots of plants that produce extra floral nectar. So extra floral, not meaning like extra flowery, extra floral, like not floral nectar. So plants will actually have little glands on their stems or where the leaves join the stem that exude nectar. They exude sugar water, basically with the adaptive value of attracting predatory organisms to drink that nectar and then maybe eat a few herbivorous insects from the plant as well. So both of those can be important. And then finally, I think of this as sort of like a strange extra floral nectary, but when plants have aphids on them, aphids produce honeydew. Basically they exude extra sugar water out of their back end, and that'll attract ants and other things to defend the aphid. But that can also be a source of sugar water or nectar-ish for beneficial insects. It's nutritionally not as good as floral nectar. And it's a little complicated because maybe the aphid is a pest, in which case you don't want it on the plant, or maybe the aphid is some innocuous little not economically important aphid that's feeding on a plant that's in your floral strip outside of your crop field, in which case, hey, that's great. They're bringing in more predators and feeding them, and that's exciting. So folks will often add nectar to their farm system with a floral strip, maybe just a fallow area, or maybe something that's intentionally planted with species that are known to be really great nectar sources. So I'm going to show you my Midwest bias. This is from some of my former colleagues at Michigan State University who wanted to create like an optimized planting list of nectar and pollen resources. So they created this big experiment. They had a grid of all these native prairie species, and then basically watched them and vacuumed all the insects off of them to see which ones are best for attracting both pollinators and natural enemies. And then they were able to create this planting list that blooms continuously from May to October, which is basically the whole growing season there, where you can have your floral strip supercharged with all the most insect-friendly plants possible. So I hope somebody's doing that for other regions in the US, but that's a cool thing to do. So that's nectar. If we move on to alternative prey and hosts, this means natural enemies need something to eat other than the pest that you're interested in having them eat, particularly when that pest is absent, right? So sometimes this happens kind of in a passive general way. If you've created off-field habitat, it may be that off-field habitat hosts other herbivores that predators can use and then spill over back into the crop field to feed on pests. But this can also be done more intentionally with like banker plants. So for example, in some greenhouse systems, this is something folks will do. So in this little diagram I've got on the right here, I'm showing you a greenhouse tomato crop that is attacked by silverleaf whitefly that feeds on the tomato. That's the economic problem that growers are dealing with. And then there's a parasitoid in Carcia sophia that's here that attacks the whitefly. Now, but it needs something else to eat, right, in a very simplified greenhouse system. So in this case, one option is actually to put some papaya plants, those are banker plants, in the same greenhouse. And then those will be, they can be infested with papaya whitefly, which does horrible things to papaya, but it completely ignores tomato, which means that's really great because it's a shared host for the parasitoid. So by having that host there, you can build up the numbers of the parasitoid, and then they're ready to attack the tomato pest when the time comes. So that's a great example of alternative prey and hosts. And I think we're going to hear about more of those from Houston later on. So now we have S and A, and then finally P stands for pollen. So in contrast to nectar, even though it also comes from flowers, pollen is mostly protein for predators. It's helping them with egg production in particular. It's often less important for parasitoids that are focused on just nectar and finding hosts, but super important for predatory insects. It's also a key resource for bees. That's bee food. An example of managing for pollen, again, in kind of a banker plant system. You know what? I forget which crop this was for, but there was this really cool study in which they were using pepper plants as a banker plant. So it was covered in a pest, and then minute pirate bugs were recruiting to that pest, and then they would spill back over into the crop that was actually being grown for sale. And then they found that if the pepper plant that was mostly there as a banker plant to hold other other herbivores. If it was flowering, the minute pirate bugs would also eat a lot of pollen and end up growing bigger and having better longevity and then becoming more effective biocontrol agents. So shelter, nectar, alternative, prey, or hosts, and pollen, that's your SNAP acronym, that's your shorthand. Great. So we also need to think, though, about larger spatial scales. And this is kind of one of my favorite things to think about is everything we just talked about. Maybe you're thinking about it like within a greenhouse or within a single crop field or that crop field and just like the vegetation that's right around that crop field. But we can actually think about this at much larger spatial scales. I consider myself, I guess, sort of a landscape ecologist. And one of the tenets of landscape ecology is that what you measure in a given location can actually vary not as a function of how management happens in that location, but what's happening in the landscape around it. So you could have, for example, two identical vineyards, or if you could take a vineyard and clone it and drop one vineyard in a landscape surrounded by other vineyards and another vineyard in a landscape that's maybe more diverse, and then measure who are the pests and who are the natural enemies in those two vineyards, you'd find really different things, not because the vineyards are managed different, just because the stuff around them is different, right? So the amount and the spatial arrangement of habitats outside of a crop field influence what's going on inside the crop field. And we can take that SNAP idea and explode it out to larger spatial scales and think about how to plan a whole farm, or even how to plan maybe with your neighbors or at large spatial scales, landscape scales. So just to illustrate this, here's an example generic landscape. If maybe we think about within a crop field, we know that insects move around maybe from the edge to the middle of that crop field. But if let's say this white line delineates like a whole farm property, we also know that organisms are moving around between different components of that property. Maybe they go to the woodlot, they go back to the crop field, they go to a hay field, different things, right? And then at even larger spatial scales, there are some organisms like lady beetles, for example, coccinellids, that will fly really long distances, kilometers, which means the amount and the spatial arrangement of habitats around a crop field matter at scales that like totally transcend the scale of individual property ownership, right? So there's processes happening that are influenced by your neighbors and your neighbors' neighbors and people a mile away, things like that. But if we think of the SNAP acronym and think about how resources map out onto real landscapes, we can think about maybe, for example, a hypothetical predator has an overwintering structure in the forest, and then it uses a nectar resource in a particular crop, and then it finds a prey item in a different crop, or maybe in a grassland or fallow system, and then a different prey item somewhere else. And you can see that all of these different components of the landscape, wink on and off in terms of their importance at different times in the year. So you can string together a timeline of those SNAP resources, and they vary in space. And how we've studied this as ecologists is thinking about landscape composition and how it affects pest suppression. So by composition, I mean the amounts or the diversity of crops and other habitat types in the landscape, maybe out to a scale of a couple kilometers. And in general, and there's exceptions to this, but in general, we think that crops that are surrounded by more natural habitats and maybe more diverse types of crop around them have better natural pest control. But it varies a lot. It depends on which landscape, which pests, which natural enemies. But as a general trend, I think this is true, right? That a landscape that has more natural habitat in it also has more natural enemies to donate to crop fields. So that was landscape composition, the amounts of habitats. We can also think in terms of landscape configuration. So configuration, which I see I spelled wrong here in blue, it says configuration. Configuration is not the amounts of different habitats in a landscape, but their size and their shape and their spatial arrangement. So you could have two landscapes that have exactly the same composition, maybe the same amount of forest, but really different configuration. For example, that forest could be lumped into one big patch, or it could be out lots of little tiny patches all over the place. And there was a recent literature review that I read where we looked at all the papers that had come out about landscape configuration and natural pest suppression and found that, yeah, the arrangement of habitats is also really important, right? Because a habitat being far away from a crop field versus close to it is probably going to matter. One of the biggest, most consistent kind of trends that we think there's growing evidence for is that fine-grained landscapes are particularly good for pest suppression. So what I mean by fine-grained is landscapes that are made up of lots of tiny little patches of agriculture, as opposed to landscapes that have fewer larger patches of crops. So for example, on the right hand here, I've got these fields are almost a kilometer across. On the left, I have a very fine-grained landscape where the crop fields are really, really small. And it can be logistically difficult to work in really fine-grained fields. But also, if you can imagine a landscape that has lots of edges and lots of different habitats, diverse habitats abutting each other, that makes sense that the SNAP resources would be distributed in ways that are more convenient for beneficial insects to access. So in general, we think right now that complex landscapes are better for beneficial insects, better for ecosystem services. They're certainly better for biodiversity. So we've, over the last long time, been kind of gradually homogenizing our landscapes and creating simplified landscapes made up of just a few crop types in big fields. I would argue we need to push it in the other direction, create landscapes that are compositionally complex, configurationally complex, have natural areas in agriculture interwoven with each other in order to maximize services from beneficial insects and also maximize biodiversity conservation in general. So one idea, I guess, that I'll leave you with here is that if we're thinking about how to manage those SNAP resources at really broader spatial scales than just within a crop field, we have to understand like what spatial scale matters. What are we talking about? And the answer is it really, really depends on the type of organism that we're thinking about. So I talked earlier about coccinella beetles, lady beetles. Some of them can disperse like 2 kilometers, really long distances. So what's relevant to them might be out like a mile away from your farm, and that still matters to them. At a medium scale, there are like some predatory bugs that maybe over the course of their lifespan, they move a couple hundred meters, but they're pretty much staying on a farm and its neighbor. And then there are like carabid beetles that there are some that really don't fly that far. Maybe in their lifespan, they only move 30, 40, 50 meters. So basically, they're constrained to a field and the margins right next to it. So the scale that we need to think about SNAP really varies depending on what kind of organism you're thinking about, particularly if you're targeting a specific pest and a specific natural enemy of that pest. All right, so I hope that gave you a useful overview of some of these concepts. And I think I'll let other folks take the reins for a couple minutes before Houston gives a short talk.
Yeah, now we are going to run a 2-question poll to that I hope you see right here. Please just give your answers. And then Nate will go over them. Courtney, does Nate see the answers to these questions?
I can see him.
Oh, you can. Okay, great. Yeah, well, how about if I hand it over to you, Nate? And we can end the poll?
Okay, great. Okay, I can still see it. That's great. Okay, question number 1 was landscape composition refers to, looks like I think I tricked you all. So the correct answer is B, the amounts of various habitats in a landscape. So landscape composition is how much of different habitats are there? What is it composed of? And it's tricky. Landscape configuration is the spatial configuration or the spatial arrangement of habitats in the landscape. So that's a tricky one because they're really related concepts and they both start with C and they both have a lot of syllables. So I totally understand mixing them up, and I have done the same thing in public talks lots of times where I just scramble my words. Great. So that's landscape composition, which again is the amounts of various habitats in a landscape. And then number two was the most important spatial scale to think about when considering habitat for natural enemies is it depends on the type of organism. Right. So that was easy. That was my last slide. Um, yeah, it really depends. Organism, I think this is a really cool concept. Organisms have different spatial scales at which they move, and they have different spatial scales at which they perceive their landscape, right? So if they can't go that far, their landscape is small. If they can, it's much larger. Great. Okay, so those were the poll questions, right? I think that's all of them.
Yes, that's all. We're going to switch now to Houston, and then we'll, or I think, yeah. Why don't we switch to Houston, and we'll, Nate, you can start to look and see if there are any questions in the chat you might want to answer while Houston's presenting, and we can also get to them after Houston's presentation. So Houston, I'll hand it off to you.
We good? Okay. And just by way of introduction, I'm faculty, extension faculty in the Department of Entomology at UC Riverside with my lab based off campus at the Kearney Ag Center, which is just outside of Fresno. And we do stuff like this and more broadly, ecologically based pest management, research in orchards and vineyards for the most part. So we're going to do kind of a crash course because we've got 15 minutes to get through this. I can give you the 2-hour version, but we'll go quick here today. So first off, bad guys, introduction to vineyard leafhoppers. So grapes broadly, which consist of wine grapes, table grapes, raisin grapes. It's a major commodity in California. They're spread throughout the state. And one of the key pests of grapes are Erythronura leafhoppers. So you've got 3 species in the state. The western grape leafhopper, that one here is in blue. That's a native insect to California. And then about 40 years ago, we had 2 other species, the Virginia creeper and the variegated leafhopper, move into California as well. They basically all do the same thing, but in slightly different ways. And as you can see, they have overlapping distributions in some places. So we're going to be focusing here on the North Coast, Napa and Sonoma County, where we primarily have western grape leafhopper and more recently Virginia creeper. So they're all about the same size. They're about half a centimeter in length. The adults have these very distinct colorations. So brown, yellow, zigzag pattern on Virginia creeper, this kind of cream and orange with these 2 noticeable black spots right behind the head on the western grape, and then variegated kind of looks almost like mottled or camouflage or something. It's got this rusty brown coloration. You can see up here, there's a lot of nice little color, but kind of naked eye in a vineyard kind of just looks like a brownish little leafhopper. Here's the nymphs. This is the immature stage. And again, they do have differential coloration. These dark spots over here on the Virginia creeper leafhopper, the yellow spots here on the western grape, and then kind of overall with variegated this orange coloration with these dark stripes on the side. They all lay their eggs right underneath the leaf tissue. Virginia creeper typically does it in groups. Western grape does it alone. Individually laid eggs, variegated does the same, but they put them even deeper into the leaf tissue. All these insects, they're pierce suck feeders. So they feed on the mesophyll of the leaf. That reduces the ability of the leaf to do photosynthesis. So you can see here on the left, this is a heavily damaged leafhopper damaged leaf. It's almost entirely white. So as they start to poke into those little plant cells and feed those, you'll get this white stippling pattern across the leaf. And in really extreme cases, you can see here on the right, dried up, crispy leaves. And that's impacting the plant's ability to do photosynthesis and ultimately to ripen fruit. So it has impacts on crop yield and quality. And also they are kind of a nuisance pest if people are in there harvesting grapes at the end of the season. These things can be in high numbers flying into your ears and nose and mouth, and it's very unpleasant. So they overwinter as adults. They come out in the spring. They deposit eggs into the grape leaves. Those eggs hatch, and you have the immature stage, the nymphs. They go through multiple what we call instars. Those are developmental stages, and then you get a second generation of adults, and they repeat the egg nymph process again, and then they go back into overwintering mode. So 2 generations, maybe 3 in the North Coast. This is all kind of driven by temperature. So there you have the insect, the pest we're dealing with. And then the natural enemies of leafhoppers, we group these into two broad categories. Predators, these are things that go in and chew up the insect. They directly consume it. So either the egg or the nymph or the adult or some combination therein. This is a list of the key generalist predators that we find in vineyards, about 90% of which consist of spiders. So they're a very important group in this system, followed by minute pirate bug and lacewings. The challenge with generalist predators is that they are generalists. So they do eat leafhoppers, but not exclusively. And they may eat a lot of other stuff that we don't care about, or in some cases, what's known as intra-guild predation. They actually might eat each other. So they're kind of two good guys taking each other out while the leafhoppers population just builds and builds. So the studies on predators in vineyards for leafhopper control have produced very inconsistent results. I still think they're an important thing to try to, conserve and enhance. But for this project, we really focused on the parasitoids, another group of natural enemy. And in this system, you have 3 species of parasitoid, all in the genus Anagris. These are what's commonly referred to as fairy flies. So they have these little setae coming off of their wings, kind of like little feathery things almost. They're one of the 5 smallest known parasitoids on planet Earth. So as you can imagine, a total joy to work with in the lab. It's like looking at a grain of pepper in a vial bounce around. So these things, they go seek out leafhopper eggs, and they pretty much almost exclusively reproduce on these leafhopper eggs. So the female will insert her ovipositor into that egg. She'll deposit one of her eggs. That egg will hatch and eat up all the contents of that egg and use that structure as a place to develop. And so you can see here, these are some Virginia creeper leafhopper eggs with all of which have been parasitized. You can see the little eyes of the developing parasitoids there, and almost like a little grin on their mouth. So they'll chew their way out of that egg and exit kind of vertically once they've completed development. And that's what you see here on the right. And then they go on to mate and continue to do this parasitism thing. And so these, because they're so specialized, we typically see most biological control programs for leafhoppers and vineyards really focused on this group. So now you've kind of got the characters and the scene setting, and now we'll talk about this habitat diversity project. So this is something that we did in collaboration with numerous growers, wine grape growers up in the North Coast, primarily Napa and Sonoma County, about an hour north of the San Francisco Bay Area. For a variety of reasons, which I won't fully go through here, these growers, like many growers in California, are really interested in developing non-chemical pest management strategies or ecologically based pest management strategies. And at the time, folks were experimenting with cover crops and hedgerows and other forms of diversification within their vineyard, and really wanted to kind of take a scientific approach to it. And so that's where our team came in and started to do this work. What exists currently, there's a couple of really nice resources out there for what hedgerow species you can integrate into your farm, and what is the bloom timing of them, and how to best establish and manage these plants, and what beneficial insects are attracted to them. But really, in all of these cases, where you do this, it really needs to be developed in a specific way that addresses the very specific needs of the cropping system, of the insect pest and beneficial insect that you're trying to enhance. So it always kind of needs to be worked out in a very idiosyncratic way. And so, the thinking was, well, we have these ideas, we have these guidelines, we're kind of experimenting with it. Can we really dial this in for the North Coast? And so the first 5 years of this study really was just a series of small pilot studies and grower exchanges where folks were trying to figure out, what will work, what flower species will work in North Coast vineyards, given the different agronomic and economic and climatic, variables that were at play here. And so we had all these little trials going on where people were doing this and that and modifying tractors, and then we'd have field days, and the growers would exchange information about it. And ultimately, what we landed on was the use of Phacelia tanicertifolia, Amymagis, and Daucus carota as a summer flowering cover crop treatment that could potentially attract and enhance these Anagris parasitoids and lead to increased biocontrol of leafhoppers. And we chose these for a variety of reasons. One is that they have an overlapping bloom sequence, so you get something in bloom throughout a majority of the growing season. And then, of course, ease of integration with vineyard management. That was kind of the whole one of the key things we were trying to do is we didn't want to run an experiment with some treatment that would never scale up. So all these things are sown in the fall. They're drought tolerant. They don't require additional irrigation, and they don't compete with the grape crop itself. So this is just a series of images showing these flowers. We put them in the row middles and alternating rows. And then as one flower would bloom, we would cut it. We would give it a high mowing before it would actually go to seed, because then it starts spreading to the vine rows and becoming kind of a weedy problem. And that would open up the canopy of that cover crop and allow the next species to come up and bloom. So here comes the Ammi majus, and you can just see the grapevines developing as we go and everything else drying down throughout the vineyard. You cut the Ammi, you're kind of getting at the end of the season, the wild carrot comes up, and then folks go and clear all that out and go in for harvest and then re-sow those flowers again. So once we had this experimental treatment set up, we went ahead and did a replicated multi-year study where we were looking at not just biocontrol of leafhoppers, but also crop vigor, yield, and quality. So again, you have instances where you might get good biocontrol, but it has a detrimental effect on the crop. And so you've got to kind of track all these variables simultaneously. So what you're seeing here, this is showing the generalist predators that were attracted to the flowers at peak bloom. So here's Phacelia at peak bloom, here's the Ammi and the Daucus carota, and then F, that's the plot with those flowers, and then the C, that's the control plot. So you had just resident weedy vegetation in there. And what you can see is that there was enhanced both diversity and abundance of these generalist predators on those flowering cover crops at peak bloom. When we looked at populations in the canopy of the vine, we actually didn't see any significant differences. So a bunch of them were attracted to the flowers, but never really made their way up into the vine canopy. Similarly, we didn't really see any difference in anagris populations in the vine canopy in the presence of these flowers. And we also saw no differences in leafhopper parasitism rate, and ultimately no differences in leafhopper abundance. So multiple years of work, and then a 2-year large field trial, and sort of a bust. But don't worry, it gets better. As we went through our data analysis, we were seeing, at these different sites where we had these paired plots with and without flowers, there was some differences in biocontrol, and these sites are spread all throughout, Napa and Sonoma County. So this is just kind of showing you approximately, where these trials took place. And this landscape diversity, Nate was talking about that at the end of his presentation, is very important for these anagris parasitoids, because while you have them reproducing on grape leafhoppers in the vineyard over the course of the growing season, in the fall, those parasitoids actually have to leave the vineyard to find an alternate host in order to successfully overwinter. The grape leafhopper remains in the vineyard in a reproductive diapause all winter in the adult stage, but the anagris have got to find some other species of leafhopper that overwinters as an egg, and we typically think these are found in these semi-natural non-crop habitats outside of the vineyard. So they've got to go somewhere to overwinter, and then in the spring, leafhoppers become active again, and the anagris move back in. And so you can imagine the timing and the abundance of these parasitoids moving into your vineyard is in part going to be mediated by the proximity of the vineyard to that overwintering habitat. So what we then did was quantify the proportion of natural habitat around each vineyard. So this is just showing kind of a land use map overlaid with some satellite imagery, and then sort of this cookie cut approach that we took to extract that information for each site and then actually use that as a variable in our analysis. And what we found is that with increased natural habitat within half a kilometer of the vineyard, we saw higher early season abundance of these anagris parasitoids. And then that led to higher early season parasitism of leafhoppers, which itself was correlated with reduced leafhopper populations later in the season. So rather than flowering cover crops, it was actually landscape diversity that really seemed to be driving the biocontrol activity in this system. And as one extra thing we did at the time is to actually go out into those natural habitats and determine what specific plant species these parasitoids were using, because not all natural habitats are created alike, and they're not just using anything that's out there necessarily. So we spent 2 years taking cuttings from more than 70 different plant species and all these different types of natural habitats around the North Coast, pulled out the parasitoids from those, and also collected all the leafhoppers so we could try to understand what is the actual leafhopper species they're using on those plants. So what we found is that blackberry, this is a known host for Anagra's parasitoids, but it's really limited to riparian areas. And so it's not always in close proximity to vineyards as we'd like. But we also found that coyote brush was a really, really useful plant for these parasitoids. And this was previously unknown. And if you know coyote brush and you know the North Coast, that thing is all over the place. It's a native plant to California, but it can persist in these really rugged, kind of disturbed environments with low water. So coyote brush was a really exciting find, as well as ceanothus and alder and a handful of other things. But really, we think in the system, coyote brush is doing a lot of heavy lifting. And we saw that by sampling every month over the course of the year, some of these plants weren't just being used as overwintering habitat. They were actually, you were actually having parasitoids emerge from those plants over the course of the entire year. So they're not just inoculating the vineyard in the spring, but they're actually serving as a source of these parasitoids over the entirety of the season. So at the end, as I said, coyote brush and blackberry, those are really the primary Anagris overwintering habitat, and they seem to utilize that over the course of the year. And going back to this figure, it's not just landscape diversity per se, but really the proximity of coyote brush and blackberry to the vineyard that seems to be driving these biocontrol dynamics. So I think I got under the 15-minute mark. Really looking forward to the discussion here. You've got all my contact info on this slide. I just want to acknowledge all of these different people that collaborated on this mega project, and I just kind of scratched the surface, and funding provided by the Van and Bosch Scholarship and the American Vineyard Foundation. Thank you all so much.
Thanks, Houston. Wow, that was awesome. And now let's run a poll. There, you should see 2 questions that Houston came up with, and we'll take a minute or so for you to answer them. Looks like that might be it. 80% of the people answered. That's great. So let's end the poll. And Houston, do you want to go over?
Yeah. So where do grape leaf hoppers deposit their eggs? Look like you'll mostly got that correct. So leaf tissue is the right answer. So they just deposit it right under the surface of the leaf. So you can, if you recall some of those images, it almost looks like this little blister kind of raised structure on the underside typically of the grape leaves, although they can be on the top as well. So right into the leaf tissue. And then what stage of grape leaf hopper is parasitized by the Anagris parasitoids? So eggs is the correct answer there. So again, those eggs are right under that leaf tissue. The parasitoid lands, it'll search around, and then it'll take about 2 to 3 minutes what she's decided she's going to parasitize it to kind of get on top of the egg, position herself, put that ovipositor, kind of like her stinger, into that egg, and then move one of her own eggs into that egg tissue. So, there are parasitoids of the nymphs, but they're really pretty rare, whole different type of insect. So we don't really see a lot of activity, but for the Anagris, it's the eggs. That's the stage that they parasitize.
Okay, great. Yeah, well, we were going to have you answer questions now, but it looks like there aren't a lot of questions.
One in the chat about blackberries and brambles getting invasive. Yeah, blackberries, this was like I said, this is a known host of overwintering host of Anagris for decades. But indeed, it's hard to expand the amount of blackberry you have in a region because, and there's been attempts to take them, put them in non-riparian habitats, give them shade, give them a lot of water. They just don't like not being in there in that type of environment. And more so, recently, well, not so recently, I guess, but they do harbor the Xylella bacteria that that will cause Pierce's disease. So, a lot of growers, when we first started this work and we were just giving kind of a background on the literature, and we didn't know about coyote brush because we hadn't done that research yet. When we said, yeah, it looks like blackberry, that's kind of the key overwintering species. And a lot of the growers were like, we just ripped all of our blackberry out because of this Pierce's disease concern. And it also is a host for blue-green sharpshooter, which is a vector of that. That's how that xylella makes it into the grapevine. So, it is important for us to try to find another overwintering species, and indeed, the coyote brush that doesn't seem to be related to any of the problems in vineyards, pest problems, disease, otherwise. So,
Yeah, well, there's another question. How receptive have vineyard growers been to this approach?
oh, I mean, very receptive. They came to us initially and said, "Hey, we're doing this stuff. Can y'all come please do science on it?" The challenge at this stage, it's like, okay, we know coyote brush is an important plant. We know having more of it is better, but we don't know exactly what the ratio of coyote brush to vineyard should be to generate a reliable result. And so, that's something that's like one of those loose ends I've always wanted to try to get back to so that we can provide more reliable guidance to growers, um, because right now it's hard without that information. It's hard to justify, you know, putting in it's just hard to do the math on what the economics of that would be. Do we need not just the amount, but also the configuration of it. Do you need every 20th vine row, take out that vine row and put in a big thing of coyote brush, or is it okay to encircle, a 40-acre field with coyote brush? And there's just a lot of questions about that. So the growers are very receptive to it. I mean, I think all growers I've ever worked with, they are looking for ways to reduce chemical pesticide use in their operation, whether it's vineyard or almonds or whatever, but they need a reliable alternative. And that's challenging in some cases because these, ecological studies take time and there's a lot of variables. So I'd say they very receptive, but there's challenges to implementing it in some cases.
Yeah, well, in our next lesson, we'll hear about how hedgerows are cost effective, and
Cool.
we love to lift up coyote brush and
Yeah.
encourage everybody to put it in hedgerows.
And
So
I,
there,
yeah.
Mhm.
Just saying, we haven't, in some of our work, I didn't present on what we were also trying to show is the stacking of ecosystem services. So coyote brush can support potentially pollinators and bat populations and these parasitoids. The more value you can show, and all of that takes research. I think that return on investment equation sort of shifts as you know more about other aspects.
Yeah. Well, before we get to some more questions, I have one that kind of ties into what Nate was talking about. How far do these parasitoids fly, these anagras?
Yeah. So, what Nate was presenting is kind of how I generally think about it. The bigger the organism, the further it can go, and then get smaller and smaller and smaller. But then you get to this point where they're so tiny, their dispersal capacity actually can become really large because they function like aerial plankton. And if you look at, these anagras parasitoids, they are tiny and like they for sure can fly. I've seen them in a still cage, still air, got to make their way around, but someone at some point I was kind of trying to describe it. It's almost like swimming or something. Like they're like an air current can probably lift these things. And we've never done. We've seen some small scale studies looking at their dispersal, within a vineyard block. But I've always wanted to use, or even I think you'd have to develop some other methods to figure out how far they can disperse, and can you collect some in a vineyard and see that, wow, they came from some source dozens of miles away or something. So it's another outstanding question, and I there's a lot of outstanding questions that remain, but
And then Nate, wind must have a influence on how insects move.
Oh yeah, for sure. Um, yeah, I think I mean a good example is there are a number of aphid pests that can be big problems like in Eastern North America, but they actually can't survive winters at all there. So they basically everything above some latitude in Tennessee or something dies. And then there are overwintering refuges like in the South, and then those aphids just kind of slowly blow in. So, you know, if there's a big uh, weather pattern that moves a whole bunch of southern air north, that's carrying aphids. That's basically how they make their way north. So yeah, pests and yeah, aerial plankton applies sometimes to pests and natural enemies. I like
Well,
that.
I was thinking about planting a hedgerow upwind of your crop might be good because that aerial plankton would blow in. Yeah, so but back to the questions, Houston, there's one about what Anagra species has been most effective.
Yeah, and I gave a quick answer, but just to nuance that a little bit. So when Virginia Creeper moved into the North Coast, and this is like a whole other project I've been working on, but you know, we knew that we had an Agris danii in that region. This is in Mendocino and Lake County now. And for whatever reason, that strain of an Agris danii out on the North Coast actually won't attack Virginia creeper leafhopper. And there's some various kind of theories around why that's happening. And so we actually went out to another part of California, found another, what we're calling another strain of an Agris danii that would readily attack Virginia creeper. And we had this large project for about 3 years where we were mass rearing that novel strain and bringing it into the North Coast and introducing it. And we saw increases in biocontrol, but it never really established in the region. And we actually this summer are doing some more molecular work to better understand, are these different species of anagris or are they just different strains? This group of insects, as I said multiple times, they're so tiny. And you know, you may read old literature that's about Anagris epos. That's the species that was referred to in vineyards for years. That actually turned out to be a complex of 3 different species, the ones that I listed in my talk today. Anagris danii may actually be a complex of 2 species. So we're still, you know, again, there's so, as you can see, there's so much more to understand here.
Yeah. Here's one. Just to recap, seems like overwintering habitat is dramatically more important than seasonal forage for parasitoids. Is that correct?
Yes, that's the takeaway.
That's good to reconfirm. Do you think other California native plants could prove good hosts for beneficials?
Yeah, I mean, if we're talking beyond the Anagris, you know, leafhopper system, absolutely. And that's what some of those guides that I showed earlier, you know, really do a great job at saying, you know, here's these native plants, here's the beneficials attracted to them, here's when they bloom. And I think that's just a really useful thing to have. So, you know, for the Anagris specifically, boy, I don't know if there's a plant left to sample, at least in the North Coast. We went wild for 2 years. I mean, I was just hiking off in every direction, trying to get clippings of this and that, and really feel like for us, you know, the coyote brush, that was a big aha moment because it if you've been in the region, it's all over the place. It just makes a lot of sense that that would be a key plant, and it's a native plant, and you know, the Western grape leafhopper is native to California. So there's some kind of evolutionary biology stuff that you can get into there. But yeah, definitely. There's other California native plants are good for beneficials. How that works in a certain cropping system is going to be very specific.
Thank you. I want to put a plug in for our beneficial, it's called Managing Pests with Predators and Parasitoids. It's in the resource list that we provided for this session today. It's a Wild Farm Alliance resource. We have to move on to the evaluation and quiz. So thank you so much, Nate and Houston, for your presentations and for answering questions. And if we have questions we haven't gotten to, we will put those together in an email and send them out. So now we'd love to get your feedback through a survey. It helps us get better at these webinars. If you want DPR and CCA credits, you have to give us your license numbers in this survey. And specifically for DPR credits, you need to answer the 10 quiz questionnaire. And there's the link to those surveys. And yeah, I think that's it. So we'll keep this open for a couple of minutes, and please consider joining our other lessons. And thank you for participating today. Thanks, Nate. Thanks, Houston.
Thank you all for hosting. Always a good time.